Good morning, everyone, and thank you so much for joining this event virtually. I'm thrilled today to be telling you about Andson Biotech and how we're enabling tomorrow's biomanufacturing by measuring what matters. So to start, I want to introduce our team. My name is Mason Chalmanzik, and I'm a postdoc at Georgia Tech and the technical lead on this project. And the co-founder of this endeavor is Andrei Fedorov, who is a professor and serial entrepreneur, including the successful foundation of startup company OpenCell. You can see at the bottom that we have a number of key advisors that have been crucial in our trajectory so far, and we're very grateful for that support. So to start, I just want to say that modern medicine can and is revolutionizing how we treat patients, but there's still critical bottlenecks really in the development of these therapies all the way from lab to market. And when I say modern medicine, I'm referring to cellular immunotherapies, genetic therapies, and regenerative medicine that are treating various forms of cancer, intervening in inherited diseases, repairing damaged tissues. And so from every stage, like I said, all the way from lab to market, we have critical bottlenecks. And first, when you're developing these therapies in the lab, you're really in the discovery phase, and you need to understand what should we measure. So what process parameters, what quality attributes are actually indicating how these therapies are going to perform, and how do you understand which ones are actually key? Secondly, once you have identified what might actually matter, you need to figure out when, where, and how do you measure it. So how quickly do you measure it? How accurately do you need to measure it? And what analytical technique are you going to use to measure these quality attributes or process parameters that are indicating your process trajectory or your therapy status as you manufacture it? And lastly, assuming that it has been a successful process of identifying critical process parameters or other types of attributes that indicate your therapy, how do you improve and control outcomes, assuming that you can measure these? And so for advanced therapy workflows that rely on living cells to produce a therapeutically potent output, generally speaking, the critical process parameters and critical quality attributes come in the form of biomarkers that are either inside the cell or secreted by the cell into the extracellular media. And these biomarkers are incredibly difficult to detect, and oftentimes you need to detect multiple of them to fully characterize these very complex and powerful therapies. And so identifying the proper technology to actually monitor that can be challenging. And so what Andson Biotech is doing is developing technologies that measure the what, when, and where for advanced therapy manufacturing. And so on the left is a representative workflow of an advanced therapy where you have living cells and also very expensive and complex chemicals mixed into a bioreactor that is then let sit to go for about 2 to 3 weeks with really rudimentary process analytics to indicate how the process might turn out. And the result is relying on endpoint assays to ensure that this therapy is safe, efficacious, and potent. And the result is high cost, high variability, and low yield. And one of the key bottlenecks in advanced therapy manufacturing is a lack of suitable real-time process analytical technologies that can characterize those things that actually matter. And what I'm referring to are those biomarkers that are in very low concentration and difficult to detect. And so what Andson has done is developed a technology that can either sample extracellular media or actual cell content directly from a bioreactor, and then condition that sample rapidly to enable real-time mass spec analysis. And mass spec is one of the most suitable analytical technologies to detect these biomarkers due to its nature of being highly sensitive and untargeted. So not only can you monitor what you know you need to look for, it also has the ability to detect things that you didn't know were important. And so by bringing this technology online, we can actually enable feedback control loops using mass spec analysis. And there's a number of key benefits to adopting mass spec as an analytical technology. First, characterizing those inputs and the batch variability to ensure your process comes out the same way each time. Secondly, of course, in-process monitoring, which is a huge void in existing sensors right now, can be improved by enabling real-time mass spec. And lastly, by monitoring these quality indicators that I'm referring to, these biomarkers that actually indicate your process trajectory, you can replace the existing endpoint assays with an assay that can actually detect these biomarkers in real time and allow you to either start your process over or cut your losses early, such that these therapies can be delivered to patients that need them on time. We really have two beachhead markets, and you can see the advanced therapies market is quite large, and the cellular therapy market, which excludes genetic therapies, which is really where we're targeting. We're targeting cell-based therapies is still very large. And then, of course, the approved therapies is a growing but much smaller field. And so first, we really can benefit these already approved therapies by reducing the materials costs as well as the quality control costs. So materials and supplies and quality control together account for 75% of the drug costs. And I'm talking these drugs are $500,000 to $1 million per patient. And our technology, due to its ability to rapidly characterize these biomarkers, can reduce QC costs and optimize material usage and improve yield. So this is a key benefit to those therapies that are already on the market. And in addition to that, therapies that are still in development, so these cell therapies that are costing over $1 billion to get FDA approval, this is a huge market segment. And really, one of the key benefits of adopting our technologies is the ability to define these quality standards for FDA approval and to determine drug mechanism of action, such that you actually have better chemistry, manufacturing, control standards. And of course, by adopting technologies like ours early on, you can optimize and scale the production process. And our technologies can reach both market segments without any changes to the approach. So what we do that makes us, I would say, different than other process analytical technologies is that we measure the right stuff rapidly. And so here I'm breaking down process analytical technologies into indirect PATs that measure correlated variables, and I'm referring to pH, oxygen, and temperature. And these are commonly used and necessary to maintain culture viability, but limited utility in advanced workflows. Whereas our technology is a direct PAT that measures these quality indicating biomarkers in real time. And so you can see that we have very small sample sizes, which is important. This is a very expensive process, and the medically potent therapeutic cannot be used for analysis. Otherwise, that's costing money and also hindering the actual therapeutic dose. Secondly, we're fast, so we can do these analyses in under 10 minutes. That enables us for real-time analysis. And then we have the ability to detect a broad range of these biomarkers, such that you have coverage over all the biomarkers that might monitor. And as I mentioned earlier, because of mass spec's nature, it is untargeted, which enables it to detect molecules that you didn't know were there, such that you can use this technology as a discovery tool to understand your process and develop better process understanding, as well as a monitoring tool to target those molecules you know that are critical to your workflow already. Okay, so I'm going to quickly go through two of our technologies, starting with our rapid media analysis for cell state monitoring. And you can see a picture of this technology right in front of a mass spec where it samples directly from a cell culture, conditions that sample, and has mass spec analysis within a minute. And so you can see here, using a fingerprint analysis of 3 different cell types, we demonstrate that our technology can detect phenotypical differences in these cells in real time, under a minute. And so how we're enabling that is that we're enabling sample treatment to enable biomarker detection. So if you were to put that cell culture media directly into a mass spec, you wouldn't be able to identify or detect any of the biomarkers. But our sample preparation enables enhanced biomarker detection. So we take that cell culture media, we put it into our novel mass exchanger, which has a few steps, including removing molecules we don't want, enhancing it with signal enhancers. And the output is the ability to detect these biomarkers with improved sensitivity and multi-component monitoring. So this is critical, as I've mentioned, that numerous process parameters in the form of dissolved biomarkers are going to indicate your process trajectory. So you need to be able to detect them all at the same time. And this technology can enable really new discoveries. So direct from culture analysis provides insight into what's going on in the microenvironment for these cells. And you can use that data to detect cell states, so differentiated and undifferentiated stem cells, for instance. And then using that same data, you can identify potential quality indicators and then confirm those quality indicators with orthogonal approaches to develop chemistry, manufacturing, and control standards and improve your chances of FDA approval such that you can actually get these therapies to market. Our second core technology is an intracellular cell state analysis that replaces a conventional sample workflow with a rapid integrated microfluidic processing device. So at the top, you can see multiple steps necessary, whereas we replace all of these steps with a microfabricated device that captures and lyses those cells and then carries out inline mass spec analysis in under 10 minutes. And you can see, using some clinically relevant human umbilical vein cells, we were able to detect 90% of amino acids and 66% of metabolites in real time. And so this really demonstrates that we have technologies capable of detecting cell state from the extracellular perspective and the intracellular perspective, providing a full picture of the biochemical status. And so I'm going to end it here and just show where we've been, where we're going, and really the path forward. And I want to just say that we're open to collaborations and early stage investors, and we would be thrilled to talk more. And you can see our contact information at the bottom. Thank you.